Today's topic is secondary cell culture. Secondary cell culture. Secondary cell culture is a technique which is also known as subculturing. Which is also known as subculturing. What is meant by subculturing? Subculturing is the process of transfer of cells from one culture media to another culture media. So the transfer of cells from this culture to this culture. So the cells are transferred from one culture to this is the first culture and this is the second culture. So from first to second media, the cells are transferred. Such type of culture we'll call it as subculturing or secondary culture. Here we need to know an important term, that is passage number. Passage number. Passage number is the number that is given or indicates number of times a culture has been subcultured. So if the culture is transferred one time, the passage number is 1. Or if it is transferred another time to another petri dish, and that culture number is or passage number is 2. So the passage number is the number that indicates a number of times the culture has been subcultured. And here a growth curve plays an important role in subculturing. The growth curve contains lag phase, log phase and a plateau phase. In this log phase is active phase where the cells are in active state. At this stage the subculturing is to be done. When the cells reach log phase, the cells are subcultured. As the cell concentration exceeds the capacity of the medium, then it's a time to subculture. The medium should be changed. The cells exceeds the capacity. Here the cells, if increases more in number, the cells needs to change the medium. Okay. Next we will see the types of subculture. The types of subculture, most commonly we see two types of subculture, that is first one is monolayer subculture, second one is suspension subculture. So these are the two methods subculturing follows. First we will see about monolayer culture. In monolayer culture, the bottom of the culture vessel, if this was the culture vessel, the bottom of the culture vessel will be having one single cell in thickness. It's a continuous layer of cells. At the bottom of the flask, if this was the culture vessel, at the bottom of the culture vessel, a single layer thickness cells are arranged. These are the single cells which were arranged in the bottom of the culture flask. Okay, and this attachment of the cells to this substrate is due to two regions, that is the surface glycoproteins. The attachment is due to surface glycoproteins and calcium ions. If these two were removed, the cells can be separated from the culture vessel. So this is the primary culture from which we are transferring it to a new medium. New medium we are going to transfer. So before uh, transferring, we need to separate these cells from the culture vessel of primary culture. So we need to remove the things which are essential for the attachment. So first of all, the dissociation is done by removing this do. The dissociation of these cells. The dissociation of these cells are done by removing surface glycoproteins and calcium ions. Now next we will see the methods of dissociation of these two, surface glycoproteins and calcium ions, how these are dissociated. So there are two types of methods of dissociation. Two methods of dissociation are there. One is a physical method and the other one is a enzymatic method. Physical method involves mechanical shaking, and cell scraping. An enzymatic method involves some enzymes like trypsin, pronase, dispase, and collagenase. These are the enzymes used to dissociate cells from the primary culture. Next, prior to cell dissociation, the cells are pre-treated with the EDTA to remove calcium. So there are two things which were essential for the attachment. So to remove that attachment of the cells from the primary culture, we use physical methods and enzymatic methods. Physical and enzymatic methods are useful to separate glycoprotein association. And before this, the cells are treated with the EDTA to remove calcium association. Next, we will see the criteria for subculturing of the monolayers. Criteria for subculturing of monolayers, we see subculturing is done at particularly between the middle of the log phase and before entering plateau phase. So in between these two phases, we see the subculturing. We have to do the subculturing. Not at all this should be done. This should not be done at lag phase. It should be done between middle of the log phase and before entering the plateau phase. So there are some criteria which we need to keep in mind before subculturing. First one is culture density. Culture density should be kept in mind when the contact inhibition starts or the cells comes nearer to one another, then it's time for subculturing. Okay. Next, medium exhaustion. Medium exhaustion. When medium becomes limited, the pH drops, which indicates for subculture. It indicates the time, it tells us it's time to subculture. Okay. So medium exhaustion is one of the criteria. Next, scheduled timings. We keep a scheduled timings to change the media, such as 1 to 4 days or 7 days for subculturing. We keep particular time period to change media or subculturing. Next, we will see the techniques used for monolayer subculture. Here there are some steps involved in monolayer subculturing. Those steps we'll see. First, removal of media. Next, exposure of cells to trypsin. Next, removal of trypsin. Next, incubation to Roundup.
The current time is 22 o'clock.
Incubation to Roundup and resuspension of the cells in media. Resuspension of cells in media for counting and reseeding. Here the cells are counted and reseeded in new culture. Next, cells reseeded and grown to monoculture, monolayer. Okay, so these are the steps involved in monolayer subculture. First, medium is removed, exposure of the cells to trypsin enzyme, where the trypsin it breaks the cells or it removes the attachment. So after that, the enzyme is to be completely removed and incubated to round up the cells, and the resuspension of the cells is done for counting, and again the cells are reseeded in a new medium where the subculturing is done as a monolayers. Okay, this we can see in diagrammatic representation. When the cells reaches the confluent stage in the sense contact inhibition, whatever I am saying from starting onwards, this is a confluent stage. When cells reaches confluent stage, the removal of medium is done. Next, after the removal of medium, trypsin is added. After the removal of the medium, this trypsin is added. After adding of this trypsin, here you can see dotted lines. This is trypsin. After adding the trypsin, the cells starts separating from one another. Okay, and these cells after separation, the trypsin is removed. After removal of the trypsin, incubated for some time. After incubation, the cells, you can see it changes its shape and it rounds up. After the cells rounds up, the cells are resuspended. Here again, after resuspension, the cells are separated with each other. These individual cells are taken and reseeded in a new media. Here you can see this on new media. You can see again the cells are reseeded. Reseeded in the sense again culturing, reculturing. Okay, and the cells are spreaded as a monolayers. Again the growth of the cells takes place. When it reaches the confluent stage, again the things will repeat. Okay, so this is the monolayer subculturing. Next we will see the suspension subculturing. Suspension culturing are mostly used for the transformed cells. When the cells selected are transformed ones or transformed cells, this suspension culture is used. This is similar to the bacterial culture or yeast culture. Bacterial culture or yeast culture. And majority of these continuous cell lines follows the monoculture methods, while some non-adhesive, means the cells which are not attached to one another, follows the suspension culture method. Example, leukemia cells. Leukemia cells. Okay, these are kept in suspensions. Next we will see the advantages of secondary cultures. Advantages, if we see, there are so many advantages in this. Propagation is faster. Process of propagation means division is faster. Next, lag period is also shorter. We need more log then lag period is shorter. Next, it results in homogeneous suspension of the cells, and trypsin treatment is not required here. So it reduces time period. No need for frequent replacement of medium. There is no need to change the media regularly. Next, maintenance is also easy. Next, bulk production is achieved. So we can produce a large amounts of cells. Okay. So these are the advantages. Next, criteria for subculturing is similar to that of monolayer subculturing. The same criteria we follow: culture density, medium exhaustion, pH we consider, schedule timings, and we also consider the purpose of subculturing. Also we need to consider. Next, technique of subculture, suspension culture. Here in this we see the cells are suspended in a culture flask which contains medium. And these are stirred with a magnetic pendulum. And next, the base of the flask, this magnetic pendulum is reached and it is stirred, and contamination is checked regularly. Contamination checkups should be done. Here you can see this is a suspension culture in a stirrer flask. In this is a pendulum I told, okay, and the cells are suspended here. On the magnetic stirrer it is kept, and this is the flask where secondary or suspension cultures are done. Okay, so this is the instrumentation of suspension cell culture. Thank you.